Hi, my name's Ryan Thwaites. I'm a lecturer in respiratory immunology at Imperial College London. I'm joined here today for the SMI's Oral History series with my postdoctoral supervisor, mentor, Professor Peter Openshaw. Peter,
Like,
great to have you with us, and looking forward to hearing some of your kind of history and your perspective about the SMI. So, um, if we could start with your background within the SMI, what led you to joining?
well, I guess that, during my PhD, it became evident that a lot of the real immunology that we were seeing in a mouse model of viral infection was happening locally in the lung. We could take out the spleens of these mice, we could mash them up, we could do experiments looking at T cells and so on. But obviously, the place where all the immune responses were initiated were in the mucosal lining of the lung. So we were studying this virus, respiratory syncytial virus, which is a real specialist in the ciliated cells of the bronchial epithelium. So I think it was obvious, when the SMI was first set up in 1987. So that was sort of the year before I submitted my PhD. It was obvious that the Society for Mucosal Immunology was going to be a great place to meet other people working on mucosal sites and with a similar, mucosal perspective of the immune system. A lot of the people at that time were gut immunologists, but so much that was being discovered by the study of gut immunology was relevant to the lung. So it seemed an obvious place to go in order to really enrich our science and to have interchange of ideas about, what's happening at those different mucosal surfaces.
Fantastic. And you mentioned the convenient coincidence, I guess, in timing then between the emergence of your personal interest in this field and in the emergence of the SMI. Do you feel like that was a something coming together in the community at the time, an increasing interest in mucosal immunology?
Yes, I think there were a lot of people doing interesting stuff, particularly in relation to, B cell immunology of the intestinal mucosa, and it was such a crucial area for scientific development and collaboration. And I remember going to some of those early meetings and just being bowled over by the comparisons between what they were discovering in the gut and what we suspected might be happening in the lung. I think the lung was really lagging behind the gut. There were a lot of great immunologists who had started to try to sample and describe what was going on in the gut during mucosal responses, but relatively few people in the lung. So it seemed very important to go along and to try to emphasize the importance of the lung from an early stage.
And do you think that that balance between gut immunology and respiratory immunology has changed over the years, or do you think that that's still a
I
focus
think
for
it
SMI?
has. I mean, it was so strongly immunology focused at the beginning, but Claire Lloyd and I in particular were constantly banging the drum of the lungs, you might say, trying to get people to understand just why the lung was such an important place to work. And you know, obviously there are two ways that pathogens get in mainly. One is through the gut because we're all obligate eaters and drinkers of, of, uh, of food and water. The other is through the lung because we're obligate breathers of air. And so, those are really the sites of weakness in terms of where a pathogen might enter. I mean that and breaches of the skin because of blood sucking insects and abrasions. But the lung and the gut are the main sites where pathogens get in. So, as somebody who's interested in pathogen immunology, I think you have to be a mucosal immunologist.
Absolutely. And you mentioned at the start the focus within SMI at its inception, perhaps really on murine immunology. How do you
Yeah.
feel that that's changed over the kind of intervening years?
I think the years I spent as a mouse immunologist were transformative in many ways. It really allowed us to study that sort of cascade of relevance, starting in the mucosa, going out into regional lymph nodes, out into the lung parenchyma in general, and then on out into the periphery, where the signal from the lung is much more dilute. But I think all those mouse studies that we did, and particularly the advent of being able to do bronchoalveolar lavage, which was really a technique that we'd been doing in human medicine for a long time. We've been washing out the lungs and getting cells out to try to help us in managing lung disorders. But transferring that procedure into mice and using bronchial lavage to get the most relevant superficial cells out of the bronchial mucosa, and also doing, lung mash cells as well to some extent. But all of that really emphasized the vital importance of looking at what was in the mucosa. And we were finding cells that we just didn't expect at all. We were using flow cytometry staining procedures that had been optimized, say, for the thymus, and we were finding cells in the mucosa that we really didn't expect to see there. A lot of atypically staining cells, not the cells that you would find in the regional lymph nodes or in the spleen. So I think it vastly enriched our understanding and knowledge of the dynamics of the response to infection. You can do different groups of mice sequentially, and you can really focus on those early responses in the lung, and then chart them through their evolution and resolution. So all of that was really important. I think it was frustrating to me that for the first maybe 20 years I was working on RSV, we had very little by way of access to studying the mucosa itself. And it was only with the advent of mucosal sampling, which you were heavily involved in, along with Trevor Hansel and others. It was only with that mucosal sampling techniques and also human infection challenge that we finally became able to really do experimental work to discover what was going on in the moist mucosal linings of the upper and the lower respiratory tract. So it was those techniques that really allowed us to evolve from mouse immunologists into human immunologists, and obviously very relevant to my background as a respiratory physician, or as a pulmonologist, as you might be called. So it was nice to sort of complete that arc back into something which was more clinically relevant.
Yeah, fantastic. It's really interesting. It's interesting that we tend to think about the techniques we apply going in one direction, so being developed in mice and going into humans. But actually, as you said, lots of the things that have been clinically developed for humans perhaps fed backwards into work that people in the SMI were conducting.
Yes, that's absolutely true. And I think also now with things like the Human Lung Atlas, we're discovering so many different cell types that we never knew were there in the first place. And I think that's making us go back and think again, look again at the fine anatomical structure of the lung, and wondering, what these cells are and how they form, ecological niches that may, for example, nurture specific B cells that make IgA. So I think it's constantly fascinating to see the way in which mucosal immunology has been developing.
Couldn't agree more. So Peter, my next question, one of the main roles really of our societies and the SMI is to host congresses and organize meetings. Can you think of an idea or a collaboration that formed through people you met at one of the SMI events?
It's hard to think of a specific one. I mean, I think so many of my friendships, my collaborations in immunology have depended on meeting people at a whole series of SMI meetings. Going back, I seem to remember going to one in San Diego back in the early days, and meeting a lot of the people who were absolutely, trailblazers in the field of mucosal immunology at that meeting, and feeling a tremendous sense of excitement that we were able to come together from all over the globe, actually, and talk about the importance of mucosal immunology and the cross fertilization, and people hugely generous because they had nothing to lose by talking to you about viral infections of the lung, because practically nobody at the meeting other than us were working on viral infections of the respiratory mucosa. So very open collaborations, very generous with their thoughts and ideas, and interested in what we were doing.
Yeah. And to what extent do you think that that was facilitated by having a separate society for mucosal immunology rather than just a national or international society for immunology more broadly?
Well, at the time, actually, the general immunology meetings hardly had any mucosal immunology in them. It was remarkable just how much everyone was fixated in studying what was going on in the peripheral blood if they were human immunologists. Almost nobody was taking local mucosal samples. Everyone was taking out blood, was growing cells out of the blood or whatever, and it yeah, I think it was remarkable. The SMI really did fill a very important niche in providing a forum for those of us who were convinced that looking at local immunity and mucosal immunity was absolutely central to understanding what was going on with these mucosal infections.
Fantastic. Yeah, and I find it amazing, as a newer member to the field, just how that is still the case to many extents. I think that there are certainly sessions at main immunology conferences that are mucosal focused, but tends to be still a minority topic. So, despite, as you say, the burden of infectious diseases, particularly at mucosal services.
Yes. Yeah,
so,
absolutely.
How do you think that the SMI benefited you and your career and your advancement?
I think it was just constantly bringing fresh perspectives to the way in which we thought about local immunity, how local immunity was being generated, the interaction between the resident cells, many of which were not conventional immunological cells. They were fibroblasts, they were structural cells, they were tissue macrophages and so on. Remember, we hardly knew anything about the whole field of innate responses back in the early days of the SMI. We knew quite a lot about B cells, and there were some great people working particularly on IgA, Per Branzig and so on, and many others. There were relatively few working on T cells, and almost all the people working on T cells were really concentrated on cells extracted from the spleen if you work on the mouse, or occasionally lymph node, or from the peripheral blood if you were working on humans. So the ability to work on the relevant cells from the relevant site was not so much in evidence, actually.
Yeah, fascinating. My next question is about thinking about the future of the SMI and what you would like future SMI members to be able to learn and to gain from membership of the society.
I think the most important thing is fostering collaboration, and doing that through being very collegiate, being very open, presenting data which is unpublished or in the process of being published, and really being able to have freedom of thought to just imagine that things which might or might not be true in the future, and working out experiments which could be designed, ideally collaborative experiments that could be designed in order to solve those existing problems. So it does seem to me that the SMI is a very dynamic, open, friendly society. And I think another thing which maybe we haven't mentioned is the role of the journal, Mucosal Immunology. When it first started, we weren't sure whether it was going to be a success. A huge amount of energy was put into that, and a lot of clever input from members of the SMI, and particularly from the editors, in pushing that forward and creating the great journal that it now is.
Yeah.
So many wonderful publications in mucosal immunology have made it what it is today, and I think that's been another great force for good in terms of the SMI, as well as bringing in income, which of course is absolutely essential for a society like the SMI to flourish.
And do you think that that connection between the society and the journal is an important way of disseminating the society's work as well? So not only for publication, but kind of
It
advancement
is.
in
It is.
society.
Yes. I mean, to a degree, there is a separation, but there is that absolutely vital, important link in terms of bringing in money. There aren't that many ways that a society like the SMI can fund itself, and with many of our academic societies and groupings, it is the journal income which is so sustaining for the enterprises of the society. And so the SMI is not alone in that at all.
Yeah, absolutely. And if I might, for my final question, what do you think are the opportunities and challenges for the future of the SMI and of mucosal immunology broadly?
I think there's big challenges on the horizon in terms of science funding in general. We're all very concerned about what's going to happen in the US with what appears to be a new administration with a commitment to try to reduce government funding, which has been so important in the extraordinary growth and achievements of mucosal immunology and also immunology in general. And I think the skepticism about vaccines, which appears to be increasing amongst people who are political leaders of various kinds. I think all of this is really, really concerning. If we're going to continue to see the improvements in health which result from study of mucosal immunology and vaccinology and infection, funding is absolutely vital. And I do really worry about the future of funding, not only in Europe but also in the US. So I think that's a big challenge. I think the sort of breakdown in relations between different countries is also a big threat to science in general. And the mucosal immunology is no exception there at all. I think those are the things which concern me most. Whether we're entering an era where there's going to be less science funding and it's going to be more closely scrutinized and controlled by those with a political agenda.
Yeah. And any opportunities that you think, or any future prospects for mucosal immunology?
Yeah, that's a very interesting question, isn't it? I think, as we've said earlier in the interview, the collegiality of the society is absolutely vital, and nurturing of early careers absolutely vital. Bringing bright young minds into the field, like yourself, absolutely vital. And trying to support and promote those careers going forward. And I think also, obviously, we need to be supportive of our journals, in that they not only allow us to give to create high impact publications, but also bring money into the society. So we really should be supporting the SMI, because that's so important in terms of the funding of the SMI itself.
Absolutely. Well, Peter, thank you so much for your time. It's been great to hear your kind of perspective on the SMI history, particularly, and to hear so many similarities really between the situation at the start of the SMI and how it continues to be, but so many opportunities as well
Yeah.
to continue the great work that society has been doing.
Yeah.
So,
Well, thank you, Ryan. It's been a great pleasure. I always
thanks
like
very much.
reflecting on the past, as you know.
Thank you.